to cut here, and we're going to cut here. You're going to do that yourself. That's part of your experiment, is to do a restriction cut of this plasmid, open up that space, and then we're going to put in the insert that is also cut with XBA1 and KPN1. Okay? And we need two different cutting sites. Otherwise, if you use the same cutting site, you only use one type of restriction enzyme, then it can go either forwards or it can go backwards. And you don't want it backwards, you want it forwards. All right. So we use two different restriction enzymes, and then we get it going forwards. Okay. So now, if we look again at the restriction enzymes on the P-low plasmid, we've got a KPN1 in the P-low multi-cloning site, but we don't have an XBA1 site. So we're going to have to engineer in an XBA1 site. So how do we do that? This is the gene. This is the green fluorescent protein gene. All right, the gene actually starts at ATG, right? That's the start codon ATG. I don't know why I have the T highlighted. It must have slipped in there. Anyway, okay. This is the ribosome binding site. And here is going to be the upstream PCR primer. All right? So this is exactly what's on the P-low plasmid, as I've written it. Okay? But if we look here at this GCTAGA, it's only one residue wrong to have a XBA1 cutting site. Okay? So we're going to switch the G for a T. Okay, this is just like Letterman. I don't know if you ever watched Letterman when you were growing up. I watched Letterman. Okay, and Letterman would come in and change the letter, and then you would have something to save the day. All right, so if we change that G for a T, then we end up with an XBA1 cutting site. And then when we do the PCR, we're going to get from here all the way down to the end of our other primer. So this is the piece of DNA that you are going to make, or that is made for you actually, in the PCR. Right? And then we're going to take that piece, and here's the stop codon, TAA. So our green fluorescent protein gene is all wrapped up in there. We've got the ribosome binding site, we've got the start codon, and we're going to put it in at the XBA1 site on the PEG plasmid. And down in here, we have a KPN1 cutting site. So we're going to cut there, we're going to cut here, and then we're going to insert this into the PEG plasmid. Okay? So what are you going to do for your experiment? I'm going to give you 100 microliters of PCR product, and you are going to run that on a gel. Now, I'm going to prepare the gel for you. The reason I'm starting here, what seems like in the middle of the experiment, is because the experiment was originally designed for a 3-week lab period, but we only have 2 weeks because I wanted to get in all 4 of the modules. It's 4 separate modules. Okay, so we will do this experiment in 2 days. In order to facilitate your time, I'm going to do the PCR ahead and give you the PCR product. And if you want to read more about the PCR process and what I just explained to you, then you can read about that in the published article. Okay, so we're going to run that on a gel, and then we're going to purify that PCR product. We're going to cut out that band from the gel and clean out our DNA from that band using the Promega. Actually, we're going to use the Qiagen gel extraction kit. Okay? Then we're going to cut the PET plasmid with the 2 enzymes. We're going to cut the PCR product with the 2 enzymes. This can be done simultaneously, not in the same tube, in different tubes. Okay? And you can do the cut, and then we can purify. We're going to run that on a gel. We're going to cut that. We're going to run it on a gel. We're going to cut out the bands for the insert and the plasmid, and then we're going to purify them again using the gel extraction kit, and then we're going to do a ligation of GFP into the PET plasmid. This only takes 5 minutes, okay, with the Promega rapid ligation kit. So we'll do, you just put that in there with the T4 DNA ligase enzyme, and the buffer has ATP in it because T4 DNA ligase is an ATP-dependent enzyme. So it needs that ATP to make that reaction happen. So we're going to ligate this. And then when we've got the ligation done, we're just going to take that and we're going to transform. We're going to do transformation. We're going to transform competent Nova Blue E. coli cells. All right? These cells are designed to take up a lot of DNA. That's what competent means. All right. So let's go from the beginning, step by step, of each of the things you're going to be doing. Excuse me. Okay, so you're going to mix the 100 microliters of PCR product that I give you. You're going to add to that 100 microliters of PCR product, you're going to add 20 microliters of DNA loading buffer, and you're going to put the whole thing into one well on the gel. All right? Then you're going to take the DNA ladder that I give you. The 1 kb DNA ladder is expensive. I want you to take 5 microliters from that tube and put it in another tube, in a clean tube. All right? And you can label it ladder if you want. But then this original DNA ladder needs to go back in the freezer so more students can use it. All right, so you're going to take 5 microliters of that, add 1 microliter of loading buffer to the 5 microliters that you put in a separate tube, and then you're going to load those 6 microliters onto the gel right next to your PCR product. Okay. The gel needs to run for about 30 minutes, and then we are going to take a picture of the gel. Here's a picture from the gel. All right. Here's your PCR product. How do we know that this is the PCR product and not this? Well, we have our ladder here. Okay, if you look at the ladder, and up here are the original wells where you loaded it. All right, so this top one is going to be the size of the plasma. Now, I don't know, maybe you can see it. It's a little bit washed out with all this light here. But up here, there's a hazy view of the plasmid. You probably can't really see it that well, but you can see a plasmid up in here. Just kind of like a light shadow. All right, but then we have this band here. Now, how big is this band? Well, this bright second band down here is 1,000 Daltons. All right, so basically this band here ran just